Cainobedes Enzyme Simulation Lab. In this simulation, you will investigate factors that affect the rate at which an enzyme works by playing the role of an enzyme. The enzyme, Cainobase, that is the student snapping the beads together, will assemble pop beads into 5 chain lengths. The active site will be resembled by your hands. The substrate, the beads, and the products will be the 5 beads you put together.
For this lab, you will need 200 pop beads, a stopwatch, plastic dish, calculator, and notebook paper.
Procedure. Assign one student to be the timer and a second student to be the enzyme. If your group has a third member, he or she will record the data and make sure that you have all the materials you need. Then place the 200 beads in the plastic dish. When the timer says go, without looking at the container of beads, the enzyme should start assembling the chains of 5 beads and continue until 30 seconds have passed and the timer says stop.
Count and record in a table in your lab notebook the number of chains the enzyme was able to make. Disassemble the chains and put the beads back into the plastic dish and repeat 2 more times. Repeat steps 3 through 5 for the additional time periods of 60 seconds and 120 seconds.
Repeat the experiment, except this time soak your hands in ice water for 30 seconds before starting each test. Construct a graph in your lab notebook to show the number of chains your enzyme was able to make as a function of time.
As we conducted more trials, the enzyme seemed to become more efficient at connecting beads. However, this may not be true for enzymes because they would remain consistent, whereas humans can get better as we practice more.
This is our data table. The ice water didn't have effect on our enzyme. It seemed that the more that our enzyme practiced, the better he got.
If the subject tapes his or her fingers, then he or she will connect beads slower.
The addition of tape to the subject didn't affect their efficiency when connecting beads, since they did about the same as when they could use their fingers. This could be due to the fact that the subject has more experience with connecting beads.
If pony beads are mixed with connecting beads, then the subject will connect the beads at a slower rate.
The addition of pony beads to the container which the subject obtained beads from had minimal effect on the enzyme's efficiency. In the beginning, the enzyme struggled, but overall performed about the same as the other experiments.
In conclusion, this lab showed how enzymes work by simulating a reaction. The person represented the enzyme, his hands were the active site, and the beads were the substrates. The different treatments were meant to slow down the reaction rate, but they didn't end up affecting it. Instead, the rate of reaction increased as more trials were completed. This was probably due to the fact that the subject connected beads faster as he practiced more, so this simulation can't accurately represent an enzyme, which should remain fairly consistent.